Hi, I'm Spencer Tassone. I'm a PhD candidate at the University of Virginia, where I work with Mike Pace and the Virginia Coast Reserve LTER. Today, I'm going to be talking about some of the results from the seagrass resilience experiment that we initiated back in June of 2020. This work takes place in the South Bay Seagrass Meadow, which is a restored seagrass meadow behind Wreck Island. Wreck Island is one of the 12 coastal barrier islands along the Delmarva Peninsula that, along with the lagoons behind the islands, make up the Virginia Coast Reserve LTER. So the motivation for this work is that there was a marine heat wave back in June of 2015 that caused an approximate 90% dieback in seagrass shoot density within the central portion of the South Bay Seagrass Meadow, and the northern edge of the seagrass meadow appeared to be undisturbed. And it's known that water temperature is cooler at the northern edge of the meadow due to the presence of an oceanic inlet and shorter water residence time relative to the central part of the meadow. And some annual summer shoot counts that have been conducted since 2015 suggest that the central part of the meadow recovered in some 2 to 4 years. Yet how the meadow recovered and why the northern edge appears more resilient remains unresolved. So the research questions that we hope to answer with this experiment were to determine exactly how long recovery takes within the central part of the meadow, as well as the northern edge of the meadow. We had these annual summer shoot counts from the central part of the meadow, but we don't have anything from the northern edge. And we also wanted to have recovery rates at a higher frequency than the annual measurements that we already have. We also wanted to know what the shape of recovery was. Is it linear? Is it exponential? Or is there some sort of sigmoidal relationship? We also wanted to know if we could determine what the recovery method was. And what I mean by that is, do we see lateral clonal growth from undisturbed areas into the disturbed areas? Or do we see more seedling recruitment within the disturbed areas that sort of propagate out towards the edge of the disturbance? Lastly, we wanted to know if there were spatial differences in these 3 questions, which is why this experiment took place at the 2 locations, the central part of the seagrass meadow and the northern part of the seagrass meadow. So as I had mentioned, this experiment took place or was initiated in June of 2020. It was one of the few experiments that we were able to get going during the pandemic. And the plots are set up in these paired control treatment blocks. The treatment sites are where we went in and removed as much of the above ground seagrass as possible. These plots are circular in shape and they're 6 meters in diameter. And a literature survey that I've done suggests that this seagrass disturbance experiment is the largest to be done to date. I visit these sites monthly from April to October for shoot density counts, and I measure shoot density at 3 distances from the plot edge, and I'll tell you why in a minute. And so let's take a look at this first figure here. This first figure is the mean shoot density for the 2 locations, the northern and central locations, and the control and treatment plots. And the way you want to look at this is you want to compare the dark red with the light red and the dark blue with the light blue. The dark red being from the central location control sites, the light red being central location treatment sites, dark blue is northern control sites, light blue is northern treatment sites. And so you can see we were pretty successful. This is pre-disturbance here in June of 2020. The following month, when we went out in July, we achieved an approximate 90% dieback, which is good considering that's about what we saw that the marine heat wave did. And we didn't see any growth after the disturbance that we created in 2020. The first growing season, we did start to see some recovery, and it appears that the central sites recovered more, but the absolute rate of recovery was very similar between the central and treatment sites. And you're also probably noticing this seasonal dynamic here. What we see is that the seagrass tends to peak in July, and as the water temperatures continue to increase into August, the seagrass start to senesce, which is what draws down the seagrass shoot counts over here. This data over here on the right-hand side of the plot is from 2022. It's this year. And what you can see is that there was much higher growth in the central treatment sites than there was in the northern treatment sites. You can also start to see that there's an overlap between the central control site shoot density and the central treatment site shoot density, suggesting that we've reached recovery in the central plots. But that recovery hasn't been matched at the northern treatment sites. And we have some anecdotal evidence to suggest that there's some erosion taking place in these treatment sites in the northern location, which is causing recovery to take longer at these sites. Figure 2 over here on the right is the mean shoot density difference between the control and treatment sites. So you can think of this as the difference between, the dark blue and the light blue, or the dark red and the light red. And so on the left here, we have results from the central meadow. And on the right here, we have results from the northern part of the meadow. And this is just another way to demonstrate that we reached recovery after 24 months here in the central part of the meadow. Whereas in the northern part of the meadow, there's still this large difference between control and treatment shoot density, which is another line of evidence saying that we haven't reached recovery yet there. Lastly, down here at figure 3, this is just a pictogram showing how recovery is taking place within the treatment plots. And so we have some data to suggest that shoot density within the plots is being recovered by lateral clonal growth from the plot edge, which is what these arrows are representing, as well as seedling recruitment from within the plots. So we're seeing, growth into the treatment plots, as well as growth within the treatment plots, suggesting that both lateral clonal growth and seedling recruitment are contributing to the recovery of the treatment sites. And so just as a take-home, recovery took 24 months in the central part of the meadow and is estimated to take 127 months at the northern edge. And again, these differences in recovery rates are likely due to erosion at the northern edge treatment sites. The relative recovery of these sites appears to be a linear function, and both seedling recruitment and lateral clonal growth appear to be occurring at both treatment site locations.